So the Response Diversity Network is an organization of people who kind of all across the world. We've got about 150 members now from lots of different countries around the world, all roughly interested in the science and application of the concept of response diversity and things like ecological stability and functioning, these kinds of questions, but particularly how response diversity might relate to these ideas. Um, and so this is another edition of our seminar series. So we have this, it's roughly monthly, but we take some breaks here and there. Seminar series where we get different speakers to present, and we try and do it in a decentralized way. So speakers will present in their own time zone at a time that's appropriate and convenient for them. So it kind of rotates around the world at different places with different hosts and different speakers. And so, because of time zones and things, the audience size varies, but that's why we put it all on YouTube afterwards, and people can watch back and comment and things like that. So that's been a really great part of the network. And we should say we're also in the middle of a big overhaul of the kind of leadership team of the network. So there's going to be a restructuring of the network quite soon. And so hopefully I know we've been getting slightly less engaged the past few months, but hopefully we're gonna, after the restructure, be able to do more stuff and kind of get going again soon. So it's my absolute pleasure to introduce Kae Natsume, sorry, who got her PhD from the University of Tokyo in agricultural science, I believe, and is now a postdoc researcher in a kind of joint position between this Japan Wildlife Research Center in Tokyo, and which is an NGO, I hear, and the University of Tokyo as well. And so Natsume-san does a lot of work on kind of pollination services and different insect pollinators and response diversity. So I'm really excited to hear what you've got to say. So please, when you're ready, go ahead and present. Thank you.
Yeah, thank you for introducing me. I'm going to share the slides. Yeah, so I believe it's showing. So thank you for having me. I'm really excited to tell you about my research. So my name is Kae Natsume, and this is my face. And just what some said, I graduated from University of Tokyo, and I'm pretty fresh and just get started. And I used to hate books, but now I love the books, and I like creating digital graphics of different animals. And my research interest is I'm interested in ecological services, especially the agriculture, and also I'm interested in insects, community ecology, and climate change. And that's why pollination service is a great topic to study, and also resource diversity is my part of my interest as well. And I'm also interested in long-term population trend of insects in their response to climate change. So today I'm going to talk about the pollination by ants. This is not related to resource diversity, but it's a fun topic to talk about. So I'll just skip you my talk. And then I'm going to talk about response diversity of different pollinators against temperature variability and different temporal scales. So why study pollinator service? It's because they're very important. Pollinator service contribute to 90% of the angiosperm species and 70% of crop species reproduction. So it's something, some ecological services that we are directly benefited on a daily basis. So here is our first half of my talk. It's about ant pollination. It's based on this study linked over here. And yeah, I just show you how ants act as pollinators of buckwheat and many various crops. So when you think about the pollinators, probably many people will think about honeybees or wild bees. And some people may know that flies can act as pollinators, and beetles as well as butterflies. And I think that many people will not think the ants is pollinator when you look at ants on the flowers or whatever. And there are actually many reasons why ants are considered ineffective pollinators, because they're really small, which means they have very small amount of pollen carried, and also they do not really contact with reproductive parts. Also, they do not move that far, which means they have really little out-cross pollen. And this one is a bit interesting, but they actually secrete this antibiotic substances, which actually makes pollen inviable, which actually some people argue is the reason why ants are not really effective pollinator. And also, ants can be villains for the plant pollination, because some species get pretty aggressive towards other pollinators, which inhibit the pollinator visitation. So there are many reasons why they're not effective. But on the other hand, there are actually some plant species known to be pollinated by ants. There are 36 plant species, according to this review in the part of the paper introduction. And also, ants are known as a pollinator for two different crop species. And one of them is mango, and another one is Jatropha. So if you like mango, it's probably because you should love ants as well. And this one is a biodiesel crop, which I'm not sure if it's supposed to be eaten. But yeah, so some crops can be effectively pollinated by ants. But what about other crops? And another question is that so pollinators generally respond to the weather variation. And if ants as the pollinators, they also probably subjected to the variable weather under the field condition, but there's very little information about how ant responds to the weather variability. And I'm studying common buckwheat, and this is the pretty much single crop that I study about for my PhD and after my PhD. And it's a pseudo-zero growing temperate zone. And it's the Adiwomas flowering plant, but like mostly like pollinators because they're self-incompatible. And they're distylous, meaning they have two different floral morphs, which is ping and drum, and they have this different floral structure, whereas ping have extrusive stigmas, where drum has much shorter styles and kind of really low level of the stigma. And it's known to be pollinated by many small insects. Their studies show it's antibiotic compatible pollen stigma. But the direct contribution to fruit set has not been quantified for ants. So objectives of our study is to test and quantify the effectiveness of ants, the common buckwheat pollinators. And we are going to do this by the means of ant exclusion experiments and also examination of pollen grains and ant integument. And then we're going to evaluate ant response to weather variation and make comparison with other pollinators to see if the response is different or the same. I study buckwheat in Nagano Prefecture. There is some inflation, but importantly, buckwheat is cultivated twice a year in this particular season, so a particular area. So you get the result for two different seasons in my result section. So here is the ant exclusion treatment looks like. We have buckwheat flower, and it's a plant. It's not really high at this point, and we made this cylinder thing from plastic folders, and we sprayed them with sticky substances to prevent ants from walking up. And then also we removed the surrounding plant, so no ants can really penetrate to this particular plant. And here is some designs. And also we did our experiments in the edge of the field because it's more accessible and much easier to do. And then we evaluated buckwheat fruit set. So after the seeds get harvested, it looks like this, and we can kind of identify the seed and wilted flowers like this, and we can calculate the fruit set, which is the same as a seed set, by dividing the number of seeds by number of seeds and wilted flowers. And we get 159 fluorescences in total, and here is the sample sizes. And this is our result. we have the ant exclusion treatment, we have seed sets for 2 different seasons. And this is our result. It's the violin plot represents the whole variation of the inflorescence level seed set, and large plot represents the mean value for different sites. And as you can see, in both seasons, in every site, ants exclusion significantly reduced the level of seed set. And the mean level of the reduction is actually 30%, which is pretty significant for the buckwheat production, obviously. And then we evaluated the pollen grain carried by ants. So there are 2 major species of ants in our area, Formica japonica and Capillus japonicus. And we just collected ants from the field on the ants feeding on the flower, and just removed the pollen grain and counted under the microscope. And this is a result for the pollen grain carried by ants. They actually carry many compatible pollen grain despite the flightlessness. As you can see, both species, for example, Formica japonica carried not many for the teamwork, but for the swarm they actually carried more than 100 pollen grains per single individual, and for the Camponotus, they carried actually greater amount of pollen grains. And because they carried much larger, because their body size is actually larger than the Formica japonica, they might be more efficient in pollinating. So ants are actually effective pollinator, and their pollination efficiency might be different between different species. And then we go to the response to weather variation. Here is our method. So we just count visually observed. It counts the number of visitors on the buckwheat flowers. And we did count it for the different groups of pollinators. And we analyzed their number of the pollinators for 3 minutes in relation to the weather data obtained for the weather station. So this is kind of preliminary result for the visitation frequency of ants. As you can see, in some season, in some sites, ants can be quite abundant. They account for more than 50% in the most abundant sites in both of the seasons. And then this is the result for June. And this is the response to the temperature, and these are the different pollinator groups. As you can see, ants did not really respond to the temperature variability, but other species respond to the temperature variability. So they are pretty stable against the temperature variation. And when you look at the response to the sunshine duration, as well as the wind speed, ants did not really respond to it either. Even though some species, especially a wind speed, many species responded negatively to the wind speed, but ants did not really respond to it. Probably because they are flightless. So from the June data set, we can say that ants are very stable against the weather variation in comparison to other species. And for the September, we also did the analysis for the temperature variability, and also know that ants do not really respond to the temperature either. And for the sunshine duration, they responded a bit weakly, but significantly to it. So ants responded slightly to sunshine and wind in September, and did not respond to anything at all in June. So the result so far is that ants are very effective, and they are actually contributing probably 30% of the whole seed set of the buckwheat at the field periphery. And they do carry common pollens, and efficiency might be different. And for the response to weather, we show that ants do not respond to weather variation at all, or they increase in cloudy and windless days. Yeah, so we say that ants are effective for self-incompatible temperate crop, and they're pretty resilient to the weather variability. So there might be other crops suggested to be ants as pollinators. Some studies suggest that coffee is pollinated by ants, and cacao, they have really tiny flowers, so they might be pollinated by ants as well. And one of the study actually suggests that raspberry and zucchini can be pollinated by ants as well. So these are the potential crops that are pollinated by ants, but there might be many crops that can be pollinated by ants in both tropical and temperate crops. However, there is actually a weakness as well, which is they're only at the edge of the field. So here the buckwheat field, and we counted the number of ants on the flowers at the edge and in the center of the field. So at the edge, there are a bunch of ants, but when we go inside the field about like 10 meters, then number of ants drops very rapidly. This is because ants nest in the field edge, and they cannot be found in the center because they cannot nest in the field center, and they do not really move so far away. So this is a weakness of ants. But in small-scale agricultural fields, ants can be really important because, for example, in our particular site, we have a average field size looks like this, which is 30 meters on the short edge, 80 meters on the short on the long side. And when you assume that ants can pollinate the 5 meters from the edge conservatively, and this is the area ants can pollinate is actually about 42% of the whole field. So, which means when the field size is small, the ants can be contributing significantly to the production on the whole field scale. On the other hand, when there is huge buckwheat fields, probably the contribution of ants will decreases a lot because of the small edge to area ratio. So yeah, that's what I said. A smaller scale field, ants are more important. And the importance of field edge is actually already recognized from other studies as well. This is a study that looks at the wild bee abundance in response to the field border density. And the higher density of field border increases wild bee abundances. And this is because they increase the connectivity and they have greater plant diversity and a seaside or ground desert. And it's the field borders and edges are good for the wild bees and other pollinators, and it is also good for ants as well. And we say that when you look at the field size distribution around the world, there are significant variation, and especially in Asia, the field sizes are really, really small in comparison to, say, North America or Europe. And in these, but also these areas are extremely densely populated, so there are actually many field and very important for the food security in the region. So this means that ants can be imported in particular scale depending on the field size of that particular region. And by sustaining these small scale field in Asia or maybe other regions, that might actually increases in sustaining the crop yield throughout the future against the weather variation. So to conclude my first half of my talk, we say that ants are important pollinators of palm buckwheat and potentially other crops, which is neglected, but they should not be because they are actually significant. And also it's important in small-scale agricultural fields, which means that it's by sustaining small-scale agricultural fields in Asia, or maybe even making field sizes smaller, that's going to increase the contribution from these kind of pollinators in the same way as other pollinators would also respond to. So yeah, here's my first half of the talk. And if you are interested to know more, you can go to my paper from this link. And this one is the second half of my talk, which is about the response diversity. And this one is still ongoing project, so I'm open to suggestions. And yeah, I'm going to talk about how temperature variation in different hierarchical levels affect the relationship between diversity and pollinator service. So ecosystem functions operate under temporally variable environment, and that is the reason why response diversity is important. Response diversity can be defined, roughly defined by species response differently to the certain environmental gradient, which create insurance, synchrony, or complementarity against it, which can be a reason why diversity is important for the ecosystem functions. And it is typically defined or evaluated for the population level change or response. As you can see from this sample work, there are not people saying that regional diversity is relevant in more than 1 year, longer than the 1 year of time scales of time scales. But we say that on the behavioral, much shorter time scale level response is also can be defined by response diversity and can be significant in the ecosystem functions. And actually, many studies use the definition of the behavioral level of response. And we say this smaller scale dynamics of environment and ecosystem functions are important because environment can significantly vary on the scale smaller than interannual scale. When you think about seasonality, especially for people from temperate zones, and also weather variation, obviously, this can be strongly varying and affect the organisms and affect the ecological functions. And secondly, also mean level of the environment cannot predict the mean level of ecological functions, meaning we need to account for the variation of the environment and the ecosystem function to accurately predict the mean level of ecological functions. And to describe this point, this is the diagram I make. So ecosystem functions provided by species for y-axis, and we have variation in environment the x-axis, and this is the species response against the environment. And we have this mean level of the environmental variation to predict the ecosystem function. While with a certain species, we have the value represented by these black dots. But when you consider the full variation, the actual ecosystem functions mean level is over here, which is higher than what is predicted from the mean value. And when the response curve has a concave function, the result gets actually opposite. They tend to have a lower level of the ecosystem function from what is predicted from the mean level. And this is what is called a Jensen's inequality, which fails us to consider the smaller scale variation. And that's why smaller scale dynamics of the environment is functional. And when you think about smaller scale dynamics, there are actually hierarchy of variability depending on the time scale that you're looking at. And we focus on temperature variability in our study because it's important environmental variation for many organisms, as well as they are especially important under climate change. And this is the mean annual, for example, we have mean annual temperature, and when we have a seasonal variation in the mean temperature, and then we have day-to-day variation, and when you break it down to the much smaller scale, we have the within-day variation. And on the hierarchical level, they have the corresponding species pool or the community defined by year level species pool, and the filtering from different temperature variations, as well as the logical or the diurnal prism filters. However, so there's a hierarchical levels. However, previous studies focus on the response personal diversity or the complementarity on a single level, that we do not know how importance of diversity changes depending on this different hierarchical levels. And also we don't know what happens when we consider all of the levels instead of just considering one of the levels. And we study buckwheat, as I told you. Buckwheat strongly relies on pollinator surveys like insects because they are self-incompatible, and they have 2 weeks of critical period during flowering seasons. So for buckwheat, we can define between day level variability and within day level variability. And for the between day level, we have insects phenological response as well as response to the daily level temperatures. And then we have for the within day level, they have diurnal rhythm as well as response to temperature. So we aim to study the strengths of VF difference comparison, the comparison between day level and within day level. And then we also evaluate the strengths of VF if they change in response to the climate change at all. So here's the materials and method. We basically use model prediction values to evaluate these different level variability and VF. And so I'm going to explain how we did it. So for the temperature variation, we have between day level. So this is the mean daily temperature change. One of the example. And we have between day level change, within day level change, which we focused on 8 to 1 p.m. period because of the flowering seasons and activities of pollinators. And for the so we have year level species for this particular season, which we define as community composition. And for the between day level, there's filters. They are filtered by date and mean daily temperature to create the between day level or like the level of species pollinator community, and we multiply them with pollinator efficiency by and consider the pollinator efficiency differences. Then we can calculate pollination service for the daily level. And furthermore, we also for the missing day level, mean hourly temperature and time of day were filtering the day levels which is full to create the hour level pollinator community to create pollination service. And these brown circles, for all of the brown circles, we estimated them from our empirical data. And for the temperature variability, we use the actual weather data for our study region from 2000 to 2025. And for the between day level, mean daily temperature looks like this. And you can see there is a trend component and stochastic component. So because there are difference in the variation, we decided to divide them by linear regression. And for the within day level, they pretty much show the steady increase across the day. So we just decided to use the raw values for the model prediction. And here's the community composition, how we define them. We wanted to define community composition as realistic as possible, but also I wanted to keep the abundance of the pollinator the same. So we first analyzed the actual variation in the community and identified 2 dominant species. One is the beetle, and two is the species of butterflies. And we took these structure and reflected them into 3 different hypothetical communities. And for the low level diversity treatment is dominated by the single species, and the medium, there are 2 species dominating the community, and the high, all species are equal in number. And we kept the abundance the same for all of the 3 communities. We also quantify pollinator efficiency empirically. This is something I've been working with so far, and it's also very exciting, but this is for the other day. And we estimated pollinator efficiency for the different species, 9 different pollinator groups. And lastly, the pollinator response. We did the field work, which is actually exactly the same data set we use for the ants weather response. But yeah, we just did the visual observation. And we used GAM with a mixed effect to evaluate pollinator response. And we evaluated pollinator response to the calendar day to evaluate the phenological response. And there we regress the we use temperature and time of day as the explanatory variable to evaluate response to the temperature as well as time that diurnal rhythm. But these 2 variables are strongly correlated. So we decided to evaluate joint response surface instead of doing the separate GAM analysis. So here's the estimated phenology of pollinators. As you can see, different species have different phenological trends. And many species actually significantly responding to the calendar day. And this is a joint response surface for time of day and temperature. As you can see, there are actually difference in the response surface as well. So here we have everything on our hand. We have temperature variability, we have humidity composition with 3 different diversity level, and we have efficiency and response to the weather variability. And here we actually did not consider the competition between pollinators as well at all, because the resource is actually kind of unlimited for the pollinator numbers. So here's the result. We shows diverse community tend to provide greater pollination service on the within day level. And we had the mean level of the pollination service estimated, predicted from the weather variability on the between day level and the within day level. And as you can see, the high diversity community tend to perform better than the other community, and the medium level actually perform not so well. And one reason of these differences between 3 groups is probably because of the efficiency difference between different communities. So here is the community with mean pollinator efficiency for different communities. As you can see, the high level of pollinator diversity actually has higher mean level of the efficiency, which is actually the sampling effect of the community diversity. Some species that are more efficient than others are otherwise not included in, otherwise not abundant in other communities. And as you can see, the pollination service was actually not significantly different between the low level and high level of pollinator diversity for the between day level, but for the within day level, their levels are actually significantly higher. And it's when you think about the median level, it's only 7% increase. But when you look at the maximum level of increase, it's actually pretty large. So we see the positive diversity effects, especially in the within day level. So hierarchical level, it's actually changes the BEF. And when you look at the stability as well, they actually has a similar trend, but actually more stronger trend, which is to say that between day level, for the trend component, they do not have a significant difference between different diversity treatment. But for the stochastic level, they have some differences. And for the within day level, they have much stronger differences across different communities. So for the stability and mean level both, the within day level has actually stronger importance of high diversity, like all of diversity. So why is that so? So in order to in order to elucidate this, we looked at the degree of complementarity among different pollinator species. So in order to do so, we looked at the variance ratio. So it's actually covariance-based method used to evaluate if the covariances between different species are positive or negative. And all you need to know is that if the value is less than 1, it means the entire community, the sum of the covariances is negative. And for the if more than 1, they are actually synchronous. As you can see, for the between day level, these the species dynamics are pretty much synchronous across different level of diversity treatment, and the high diversity treatment was not complementary at all, even in comparison to other communities. But on the within day level, you can see that high diversity treatment has tend to have negative covariances in comparison to other communities, and they're pretty much the only communities at the hierarchical level with negative covariances. So this actually shows that complementarity contributes to the importance of diversity in the within day level. And this was because of the response diversity difference on the different hierarchical level. So for between day level, we show that the phenological response were different across different species. But when you actually look at much more closely, you can see that 9 out of 6 species have a declining trend over the flirting seasons, which means that they have a strong synchronous dynamics when it comes to the between day level variation. On the other hand, within day level, well, temperature variation is obviously very different, but also for the diurnal rhythms, some species gets more active in the morning, and some species gets more active in the afternoon. Some species gets more active during the day, and they have less diversity of responses, which I haven't evaluated, but responses are highly diverse within day level, which created complementarity, which leads to the higher importance of diversity. But when you go back to the previous result and look at these variation within the communities, so there might be a strong context dependency when it comes to the community diversity effect as well. And these are direct, because it's a model prediction, it's directly caused by the variation in the temperature. So in order to assess the context dependency of these diversity effects, I looked at the mean temperature and the temperature variability and how they influence the level of pollination service and stability. So here, here's the analysis. I just did the linear regression using mean temperature and temperature variability as a explanatory variable. And yeah, this is the result. So the mean temperature tends to increases the pollination service for low diversity community. As you can see, it increases rapidly, I think. Wait, I think these labels are the opposite, but low diversity level is actually increasing rapidly in response to the mean temperature. And it's the same thing was happening for both the between day level and within day level. And for the temperature variability, they do not really respond to it. So higher temperature level in the both level, importance of high diversity community or the advantage of high diversity community is actually declining. And for the stability actually decreases in response to the mean temperature as well as the temperature variability, and mean temperature decreases stability, especially in the within day level. So, but they decreases stability for the high diversity treatments, especially. So this way, they also decreases the BEF, positive BEF effect under like high mean temperature. And predictably, in response to climate change, the importance of community diversity declines as well. This is the mean level of pollination service predicted from the projected value of the temperature variability in near future. And when you look at the between day level, for example, high diversity treatment has a increasing trend towards the different level of the warming scenarios, and the medium level is kind of most cost. And for the high diversity treatment, they actually stayed pretty much the same for the between day level. But within day level, the low diversity treatment also increases, and high diversity treatment a little bit increases, but the increase is not as significant as the high diversity treatment. And for the both levels considered, it looks like this. So they both increases, but the low diversity level responds more rapidly to the warming scenarios. So which is kind of like a reflecting maybe more strongly on the within day level trend. So this is because dominant species become more active under warmer conditions, which decreases the importance of having diverse group, but instead of like high diverse group, but it's more important to have the single species that respond positively to the warmer climate. So yeah, this is kind of like a case study using our data relevant to our region, but I'd like to analyze if our system is special. And first of all, we say that the advantage of diverse community decline in the future in response to the warming. But yeah, is it general? And I think it's somehow general to the crop pollination system because dominant crop pollinators are European honeybees in many regions, which normally respond positively to the warming. Well, it depends on how much warming that's going to happen, but they actually prefer warmer weather. We also estimated the response surveys for the honeybee using our own data sets as well. And they show pretty much kind of a similar response as our dominant species we use. So we can say that under climate change, the abundance of dominant species might increasingly limit the pollination service in many systems. And the important diversity, when you compared in between site scale in the future, that's the importance of diversity is going to decrease. And yeah, so this is probably one of the probably general. And we say that diversity is important within the level. And this also can be context dependent when you think about how temperature variability changes on the hierarchical level on different regions. And when the data variation is not as large as our system, that's going to also decreases the importance of diversity on the within data level. And for the pollinator response, we had a sequence phenology, but maybe that's because we did our observation in September, which is the autumn. And maybe when you look at some crops like pumpkins, which is the summertime crop, and probably there's gonna be more complementary phenological response because during summer. So to conclude our second half, I'm going to so in realist communities, the presence of positive BF and the strengths of complementarity depends on the hierarchical level and number of variations you're looking at. And we say that we consider the multi-hierarchical level as important for the understanding of BFs under different environmental variation as well as for the predictions. Here is to acknowledge people who contributed to the field work as well as the farmers give me the permission to study their farms. And also I'd like to acknowledge my PhD supervisor, Tadashi Miyashita, and people from Laboratory of Biodiversity Science and Department of Ecosystem Studies in University of Tokyo. So here's my talk. Thank you for listening.
Brilliant. Thank you very much, Natsume-san. That was really, really interesting talk. Lots of very cool stuff there. Um, cool.
Thanks.
So we've got plenty of time for some questions if people have questions. I'm going to say again, if you don't have to unmute yourself and turn your video on. If you want to ask a question, you can write it in the chat and I'll read it out. So I have plenty of questions, but I'm going to start with one that's in the chat. So Veronica Zapata gave a comment in the chat, said, sorry, I have to go. Great presentation. One question. In the experiment that you presented at the beginning of the talk, in which you wanted to evaluate the effect of ants as pollinators by removing other surrounding plants and using the sticky substance, how do you know that the observed effect is actually due to the removal of ants and not to a reduction in flower density? Good question.
Yeah, thank you. Good question. Actually, I've never got that question before some reason. So it's actually a great point.
Yeah.
So yeah, might be so we didn't test if that's because of the reduced density. Oh, there's so many animations. But we believe that so we did an experiment in the beginning of the flowering period, which means
Hmm.
that buckwheat probably have done growing already, and they're kind of like they have all these resources of their plants already.
Hmm.
So we believe that removal of the other plants would not really affect the buckwheat that much, even if there's the effect at all. So that's yeah one reasoning I can think of. But yeah, it could be because of improved growth and resource amount by removing. We couldn't uh, we couldn't prove that's yeah purely because of
Sure.
that. Yeah, thank you for that.
Yeah, cool. Great. Thank you. And I guess also while we're talking about that, the ant part of the talk, which I thought was really interesting, by the way, I didn't know very much about ant pollination. I'd kind of heard that it was a thing, but not I didn't know very much about it. So that was great. Um, I was wondering about, you showed that figure at the end with the amount of the different sizes of, yeah, this one of the field sizes across the world. I was also wondering about the temporal trends in this data
Mm.
and whether,
Yeah.
because I imagine in some parts of the world, field sizes are getting bigger because, big companies are coming in and they're, turning smallholder farms into big agricultural
Mm.
fields.
Right.
Whereas in a lot of places, I imagine they're also probably getting smaller, right? Like, I guess this is maybe the case. Might this be the case in Japan? I was going to wonder about your field sites. I was wondering about things like land abandonment and what this means for the sizes of agricultural fields in Japan.
Yeah. So thank you for that. That's really important point. There's actually available data for Japan
Mm.
in regards to the field sizes change across years. And also there's some reason they have this field margin um length or the density probably data set available in the Ministry of Agriculture and
Oh,
stuff.
cool.
Yeah. So we're still not sure how they are calculated, but we see this declining trend in the field margin length
Okay.
or the density. And also Japan went through this huge field expansion era or like
Mm.
field kind of like hiding them hiding the field up
Hmm.
from much more like rectangle into kind of rectangle shape,
Yeah.
and that's happened around probably like I think in 60s.
Okay.
But yeah, actually there's actually Japanese agriculture is going through little changes throughout the year, which changes the field sizes and changes the complexity of the field shapes, which actually
Mhm.
probably reduce the field margin to the area ratio. Yeah. So yeah,
Cool.
that's probably gonna be doing something to the pollinators as well.
Yeah, the
And
relative contribution of the ants pollination, right?
yeah, exactly.
Yeah.
And for the field abandonment, I haven't thought about the impact of it, but the buckwheat, when you're speaking of buckwheat, they're actually cultivated in many abandoned fields because they're very easy to cultivate, and some um, some farmers can get the subsidy from the government for cultivating
Mm.
something in somewhere.
Mm.
So yeah, I'm not sure, but yeah, I'm not sure what specifically we can say about the field development and pollination of buckwheat. So yeah, that's actually something to think about, I think. Yeah, thank you.
Great, thanks. There's another question in the chat from Kishin. He says, "Thank you for your interesting presentation. Could you tell me how to define pollination efficiency, and does it depend on the insect body surface area? So do
Yeah,
larger insects, carry more pollen, basically?"
yeah. So thank you so much. Yeah, you're right about definition of pollinator efficiency. There are actually many different ways we can define efficiency, but in our particular study, we define pollinator efficiency by directly observing the probability of fruit set per visit by different pollinators. So it's it's not a trait-based metric, but more like direct observation result and some statistical analysis. But also, some people would define pollinator efficiency by looking at how many pollen they can deposit on the stigma, or yeah, it's either look at the single visit stigma deposition of pollen or looking at visit per visit. So that's kind of a major way. But yeah, there are actually many ways people like creatively define pollinator efficiency for sure. Yeah, thank you.
Great, thank you. I'm just looking at my notes here. I've got lots of questions written down. Um, I really liked your joint time of day temperature response surfaces. I thought they were really cool. I didn't quite catch, did you use response diversity or these kind of responses directly as a predictor in an analysis? And if so, how did you kind of condense these these two-dimensional surfaces into a single value for that?
Yeah, so I didn't do use didn't measure response diversity per
I
se,
see.
and
Yeah.
I'm not sure how to do it, to be
Yeah.
honest. That's like one reason, and also we want to yeah I just do not know like how to do it,
Mhm.
and also I'm using these response surfaces to predict the actual value of pollination from
Mhm.
the actual temperature variability. So it's like you don't really have to go through resource diversity, but you can just kind of directly predict the pollination service itself.
Yeah.
So yeah, so I don't know how to do it, and I didn't evaluate them because I just wanted to see if diversity relates to ecological services. Yeah.
Sure. Yeah. No, thanks. That's interesting. I can send you maybe a paper that I've seen that would be relevant for that. So
Yeah,
yeah,
thank you.
cool.
Yeah, yes, please.
Yeah, yeah. But no, I thought it was really cool. So I wanted to ask also about so it seemed to be that a lot of the time it was the high diversity treatment and also the low diversity treatment that were having the kind of strongest effects on, pollination efficiency and these kinds of things, right? And
Hmm.
often it was the medium value that was kind of the lowest, as you see here. And so I guess I was wondering what's going on. So you think it's so this is the pollination efficiency here on the so you think it's driven by this second figure here, this pollination efficiency, or
Yeah.
yeah.
Yeah. I think it's because of pollinator efficiency. So
Mm-hmm.
we have this estimated pollination efficiency for
Ah,
butterfly, which is really low. And so sadly they were not really efficient. So that's
yeah.
dragging the level down.
I
But
see. So yeah. Okay.
but for the stability, they're actually not doing
Right.
really bad.
Right.
But I mean, what is the stability for the within the level for the actual production level? So
Mhm.
it's like probably yeah, it's it's the butterfly not doing
Yeah.
really well.
I see. That's interesting. No, that is interesting because it's.
Yeah,
Yeah, I mean it's all this stuff about dominance and evenness and things like that,
yeah,
right? Yeah, actually that's kind of what struck me when you were showing the plot kind of defining these different diversity levels was that yeah, you could almost look at it as a continuous variable of like
mm,
evenness,
yeah,
right? And then it seemed very very related to evenness the way you were defining it. I thought it was really interesting. Yeah, yeah.
yeah.
Cool. Um, we've got a bit more time for questions if anyone else in the Zoom has any questions. If not, I can consult my notes. Um, well, um,
Oh,
oh,
hi,
there's a,
Orlando.
oh, yeah. He's thanking you for the talk. That's great.
Thank
Um,
you for coming.
let me see. I did have more questions, but those I've written down so many notes, it's hard to find them. Oh, I've got a very very basic question about the something you said right at the beginning about the ants maybe not being good pollinators because of antibiotic secretion.
Mm.
I was gonna just ask, do you know if that's how species specific that antibiotic secretion is? Is it meant to be like that it affects the viability of all pollen, or is it just, or is it that these antibiotics are quite specific to specific plants, do you know?
So I think I'm not sure about the plant pollen response difference between different species, but I heard so one of the study evaluating this looking at I think the pollen viability response to the substances created by different ant species from tropics and from temperate regions, and they they show that how ants in tropical areas secrete more antibiotic substances, which is detrimental to pollen viability. So we certainly know that there might be species difference when it comes to the ant size, but I'm not sure about the plant side. Probably there should be difference, I think, but I don't know. Some plants would be resistant. But probably some like plants actually pollinated by ants should be resistant to these substances, but I'm not sure if anybody has looked at if they are.
Yeah, that'd be really interesting to
Yeah,
know. Yeah. Cool.
yeah, it's interesting.
Great. Well, thank you very much. We're on 10 o'clock here in Japan now, so I guess that's the end of our hour. So thank you very much, Natsume-san, for that really interesting talk. Lots of very, very cool work. Yeah, please continue doing that work, and I'm looking forward to seeing it develop in future as well.
Yeah, thank you so
Um,
much for inviting me again, and
yeah, of
thank you for
course.
all the questions for
Yeah.
everyone.